All right, why don't we get started? So, good morning, good afternoon, everyone. Today our agenda is on the screen. Well, first, I want to sort of preamble that, as usual, our meeting will be recorded, and so we plan to post this on YouTube. If anything occurred during the meeting that you feel uncomfortable about, we can edit it out. That's fine. Today we plan to have a session dedicated to SLAP2 analysis. So this is, as you might remember, we have collected synaptic activity data using a tool developed by Kasper Podgorski, who is here, Kasper Podgorski group, with lots of help from a lot of scientists in his group, including Made, Andrew, and Mani, who is here, and many others. Today, Manav, Punekanti, and Suray, I'm not going to try to mispronounce your last name, I'm sorry, and are presenting each some analysis that was done on the pilot SLAP2 data that we collected in the fall. First, I want to preamble that they're both bachelor students, and so it's amazing actually that they are today presenting at the start of a career in the very open science fashion. So I want to thank both of them for their courage to do that. And so that set you up for success for the rest of your career. I think Manav will start. Is that right? Okay. So why not you take it away?
Amazing. Thank you so much, everyone, for the opportunity to share my work. It's just a bachelor's dissertation, so it's cool to be able to share it to such a big forum. I'm just going to try and figure out how to share my screen. Okay, cool. Now, if I go into presentation mode, will you still be able to see that? Is that visible?
Perfect.
Yeah, we see the PowerPoint.
Okay, great. So I was predominantly looking at the receptor field block of the SLAP2 data. And what I sort of started with as a sort of big picture is we have a pretty reasonable understanding of retinotropy on the sort of whole cell level or even the sort of neuropopulation level. A lot of work to show, you know, I'm sure all of you are very familiar with this, but just very briefly, we can see functional localization of like not just in V1, but also in higher visual areas. You have orientation tuning, strong receptor field structure, and in mice specifically, we don't see the sort of orientation columns arranged in the sort of like serial manner that are very typical of carnivores and primates. And then what sort of complicates this picture is when we start to look at a single neuron, even before we go to the sort of dendritic and spine level, we already know that there's a sort of internally heterogeneous tuning structure in the receptor field, which kind of raises the question of what are the sort of nature of the inputs that generate this topography. So on the spine level work, I really enjoyed this paper by Jia et al. It's a very beautiful kind of representation of how we sort of traverse across various dendritic arbors. We see a very wide range of sort of orientation selectivity and even two dendritic segments that are right next to each other in sort of dendritic space can be tuned for vastly different sort of orientations. And what we can kind of take away from this, given that it's sort of not as precise as a like fast glutamate sensor that we have for the SLAP2 data, is nonetheless we can kind of infer that there is some sort of meaningful sort of biological information encoded at this very fine spatial scale, but it's a limited field of view. And with the sort of SLAP2, we have the opportunity to try and find out some broader principles that govern dendritic input organization, not just within one dendritic compartment, but across the dendritic compartments of one neuron or even many neurons. So that's sort of where I started off. This is the kind of setup when I was working on this project a couple of months ago. This is a sort of sum total of data that was made available with the glutamate sniffer. I can imagine some of you be worried that we have like all of these nice subjects over here and then one really weird mouse, which is the interneuron knockout mouse. But I felt comfortable kind of including it in this analysis because I did leave one out for all of these and it didn't really meaningfully change the results in any way. So we all in all have a pretty like rich data, like a huge number of ROIs. I will get to why, like, even though we only have a handful of mice and a handful of valid sessions, the fact that we have so many ROIs is very useful for particular kinds of analysis that I'll get to a little bit later. And oh yeah, if you have any questions, feel free to ask it at any point.
An ROI is a synapse in a given recording, an extracted synapse within one recording. Is that how you count ROIs?
Yeah, so for legal purposes, I can't call them synapses, but we can sort of infer that they are most likely synapses on one dendritic arbor. Again, when we say one imaging plane, I take that as sort of like I can't legally say that that's one dendritic arbor, but it's fairly likely to be, although there's always a possibility that, you know, when we are imaging, well, not we, but like when Allen Institute images one imaging plane, they're kind of actually cutting across two different arbors that are not even like of the same neuron. But I kind of have some insurance for that I'll come to later in my presentation, where I feel pretty confident in saying that the data that I've looked at in this particular project can kind of safely assume that one ROI is one synapse and one like DMD1 imaging plane is a dendritic arbor, or at least one segment of a single neuron. So this RF mapping method is completely derivative of the code that Carter Peen very kindly shared and made available. I changed it a little bit. But the idea is that the delta F over F traces were all pre-computed and included with as part of the NWB files. And I chose sort of tentatively, this is again, it's quite nice that we had such high resolution images on the dendrites. It means that I can choose quite a high threshold. So I started with my analysis at Z greater than 3.5, but in the end I ramped it up to 4 and still had a huge number of like really highly spatially selective synapses. And what we could kind of see as we sort of did this, so this generates, I'm not sure if you've seen them before, but this like 7x7 grid of like binned receptive fields, and they kind of span 3 broad categories that I filtered the data into. The first is we get these like really sharp single hotspot ROIs, and then we get these ROIs that have these like distinct hotspots on them. So
sorry, quick question for the single hotspot. Are they cross-validated graphs?
Cross-validated against
is this cross-validated, or is it possible that you've just got some chance events happening there and simply have this as a high plot, or is it something
I see what you mean. I see what you mean. Yeah, so we generally pick ROIs per session, right? Because it would be invalid if I have the same mouse between sessions, sometimes with like large periods of time between sessions, for me to then average across those sessions to try and see this. I don't know if I'm choosing the same neuron every time. So instead what I did is, you're right to say that some of these ROIs might be artifactual, but when I pool across all of the mice and all of the training sessions, because for a lot of the topographic analysis, I'm not actually concerned with the binning itself. So this is a sharp hotspot, and it's but if I average across all of my sessions, I'm actually looking at the entire z-scored map. So if there are sort of artifactually sharp single hotspots or anything like that, they should be flattened out as I sort of pool across sessions and so on. This filtering stage is really mainly just to exclude one specific kind of ROI, which is these unlocalized ROIs, which would just add a huge amount of noise to any analysis where I'm trying to look at the subset of ROIs that do have at least a chance of being spatially tuned, right? Does that satisfactorily answer your question? Or?
It does answer it, yes.
Sorry, it doesn't, or?
Does it? It does, it does.
Okay, yeah, okay, perfect, perfect. Good. So when we sort of average across, when we sort of do this and we filter, this kind of addresses your question as well about how plausible are these sort of filtered ROIs, and you can kind of see that there's this pretty large distribution of receptor field sizes, but almost all of them fall into what we would expect for V1 RFs, which I take to be quite an encouraging sign. And also to address your point of like artifactual ROIs, I excluded all ROIs that fell below a particular delta F over F threshold, 0.3. So hopefully we only have like really high quality ROIs that we're feeding into our analysis in the first place. And what we can kind of see from this is that our receptor fields do have spatial selectivity, but they're very wide in the extent to which they're selective. And we're already picking a subset of selective ROIs to begin with.
Sorry.
I then proceeded to, yep.
How do we define the size of the receptor field here? So what exactly did 2 sigma mean here? Do you fit a Gaussian to this activation map you showed the slide before?
This one. So I think for the RF analysis that uses the binning, so that would be the noise correlations that I come to later. We aren't actually evaluating the RFs in any kind of like absolute measure of size. It's just relative. As for over here, how we've determined the size, I'm not actually 100% sure about whether it's an absolute size or whether it's relative to a median RF size. That's something I'll need to like check and come back to you next time. But this is just really to illustrate the spread of potential like receptor fields that we have relative between them. But when I then looked at retinotopic gradients, so any kind of classical retinotopy between in the way that we described when we looked at the sort of neuronal population, I didn't really, I can't say that there wasn't any retinotopic gradient, but the analysis was certainly underpowered, where there were very few significant session planes to begin with. And even when there were significant, they had relatively weak linear retinotopy. So just to clarify what I actually mean by retinotopy over here, I just pretty simply did these spatial fitting where a response to a particular visual bin is a function of the ROI's X position and Y position. And instead of like thinking of the ROIs as if they have one RF center, I just looked at whether the responses to each visual location changed smoothly across the imaging plane. So if we expected to see a retinotopic gradient, ROIs on one side of the imaging plane would respond much more strongly to that part, to one part of visual space, and ROIs on the other side would respond to a different part of visual space, and so on. And I just did R squared across all of the visual bins and then shuffled the ROI positions to see if that would break the sort of spatial predictability. And really, for most of these planes, it was not any stronger than expected by chance. Yeah, sorry.
Just want to clarify. So here, you are taking all of the receptive field across one neuron that is recorded, and you're looking at the distribution of those receptive field onto the screen. That's right?
Sorry, in this one in particular, I'm regressing the ROI position against the RF. We created those receptor field plots that I showed in the last slide. All I'm looking at over here is whether, as I traverse across the receptive field, whether I'm also smoothly traversing across dendritic space as well. And the way that I'm evaluating that is I'm just shuffling the RF locations against the dendritic locations and seeing if our actual data is better than that kind of chance shuffle, and it isn't for most of the planes.
I think it would have been helpful in here to have an intermediary slides to introduce those steps.
Yeah, okay, sorry.
No worries. I'm just sort of providing feedback.
No, thank you.
I have a question as well that I'm curious about.
Sure.
So you mentioned that you're shuffling I assume you're shuffling the ROIs across specific planes. Are you accounting at all for, say, the fact that ROIs might be on different dendritic branches, for instance?
So you can see that over here for these tests, I've only shuffled ROIs within plane. It would be invalid to shuffle like ROIs between planes in this instance. I take all of the ROI recordings just for one session and I shuffle them against their sort of associated like RFs. And I see if this shuffled orientation, that's what I take to be chance. And then I compare that to the actual recording and see if the actual associations demonstrate any linear retinotopy over there. And then this is just a representation across all of the, like when I do this across all of the session planes, the R squared is an indicator of the strength of association, whether as I move across dendritic space, am I moving across RF space in any kind of linear fashion? And I found that to be underpowered. Does that make sense?
I see. So that all that makes sense to me. What I'm saying is that in a specific DMD plane, at least from the analysis that I've done and the visualizations that I've had, it looks like there are ROIs on different dendritic branches, and that's visible. And so when you say that this property is varying linearly across dendritic space, and that's what you're trying to evaluate, it would make sense to me.
I see what you mean.
Yeah,
I see what you mean. So I'm not actually moving linearly down a particular dendrite if the ROIs are sampled from different dendritics. But yeah, I think that's valid. The problem is, I think, firstly, I couldn't really come up with a great method for looking at just small segments of these ROIs. So what I did later on, which I'll come to, is I overlaid these ROIs on top of the mean red channel that was also provided with the data set. And I tried to evaluate whether these ROIs are biologically plausible. Are they actually representative of moving down the dendrite in this way? And I find that this was sufficiently true that I could run the analysis. And some of my other analysis would be like, it kind of doesn't depend on this linearly traversing down one dendrite in order to be valid. But I agree that this is a limitation of this approach to evaluating kind of topography in a linear fashion, which is why I say that it's just underpowered. I'm not saying that it's not there. It's just, it wasn't really in the scope of my analysis to address this in its sort of totality. And I would say the same for orientation tuning as well, where this is sort of just a standard polar plot. I don't think I did orientation tuning justice in my project, because I'm sure Surrey will have much more sophisticated takes on this. I didn't do OSI or circular variance. This is just a simple circular regression of orientation that showed, this is just one of my sort of stronger neurons over here, that shows like a degree of cardinal bias. But generally speaking, it was sort of mixed, the orientation tuning, and I couldn't kind of identify for orientation tuning, again, any sort of like strong retinotopy in sort of the, again, when I regressed against RF, I couldn't find any sort of strong relationship between RF and orientation tuning. Which is to say that the sort of hypothesis here was, could it be possible that two ROIs that encode the same region of visual space share some sort of like underlying similarity in their orientation tuning? I'm going to rush ahead because I don't have much time, but I didn't really find anything significant here either. And this sort of brought me to look at noise correlations as an avenue of sort of circumventing a lot of the problems that all of you have raised already, actually, right? Which is, it's difficult to figure out how to isolate specific dendritic arbors in a way that doesn't leave your data like really underpowered for analysis, because there's only a handful of imaging planes where when I overlay the ROIs over the red channel, I can see very visibly and confirm that this is just one dendritic segment. This is just one part of one dendrite. And noise correlation.
Quick comment. I don't know why you use the red channel for this. The red channel should be very dim in these recordings, and the mean of the green channel is much better for seeing the structure than the red channel specifically.
You know what it was? I think I was like quite naive coming into this, and I didn't really know like how to pull out a mean from the green channel necessarily and represent it structurally. Whereas a structural representation of the dendritic structure in with the red channel was already provided with the data. So I just used that in combination with the single ROI data and overlaid it. But that's a great direction. If you know, one of you could sort of teach me how to do that, I would appreciate that very much. But
yeah, if you're still working on this, then I think a thing that we should do is sit down and share some analysis repositories and maybe a paper on sort of that goes into a little bit more detail on how we've analyzed this data in other settings. Which apologies for not having shared that before. Yeah.
Yeah, no worries, no worries.
Not this specific data. Sorry, this style of data. Other data collected with the same instrument. Yeah.
Okay, yeah, that sounds great. So
Not that Manav had access to the NWB files at the time. Now we have shared the raw files, and so there are more analysis that can be done on both assets. I feel like, yeah.
Okay, amazing. I mean, I'm kind of enthusiastic to push this further and develop my understanding as well. But so noise correlations I looked at to kind of circumvent some of these limitations, and it immediately kind of exponentially increases the richness of the data, because with the noise correlations, you can not just compare, across one dendrite or across one session, but rather you have a relationship between every single ROI combinatorially. You can kind of compare across all of them. And I feel like this is a neglected method, at least in the literature that I saw, because noise correlation is often dismissed as this kind of bandwidth limiting factor, because when we have a noise correlation, that means that there's a sort of like our sort of capacity to code sparsely is compromised. We have more interference. We have fewer bins for our sort of analog coding down a dendrite. But I read some really cool papers on how noise correlation actually reinforces behavioral readouts, which I find to be quite an intuitive idea in the first place. And I thought maybe we can use this as a way to evaluate synapse relations with one another. And I kind of started off by asking, do we even have sort of like positive noise correlations across all of the synapses of all of these neurons, of all of these mice? And we have a pretty strong like noise correlation, a fair bit of variance across them as well. But generally, the noise correlation of the entire data set is positive. And I think it gives us enough leeway to compare noise correlations between different parts of the data set and get kind of useful information out of it.
Before you get too deep into this, can I ask about the previous section?
Sure.
In general, did you find that the spatial receptive fields onto a given neuron are all in like roughly the same place, the ones that are localized, or did you find them to be diverse?
I found them to be diverse. Yeah, I found them to be diverse, and I think I have some cool charts to show you as to like exactly how diverse.
You'll get into that. Okay.
Yeah, I'll kind of try and bring it back in a little bit.
Okay, thanks.
So the noise correlations are in toto positive, and the sort of simplest thing that we can kind of expect is the physical distance predicts noise correlations well, right? Because if two synapses are close together on a dendrite, they probably received at least some amount of shared input, even if that's just glutamate spillover when between two neurons. And so we saw like a strong relationship between like noise correlation and physical distance on the dendrite itself. But where things got interesting is the sort of right panel here is showing comparing near distance, like I binned the ROIs into near distance and far distance. And we can see as as I said in the last slide, that there is like a relationship where sort of closer ROIs have a higher noise correlation than distant ROIs. But high receptive field overlap ROIs have much stronger noise correlation than low RF overlap ROI. So we have these sort of like two dimensions across which noise correlation is, relating to ROIs. And it's this one that I'm like more interested in because it implies some functional organization of dendritic space based on.
This is a case. Oh, sorry, someone else.
Sorry, yes. I just wanted to ask if on which stimulus you compute the noise correlations, because it's in principle the receptor fields are also computed based on correlations. So I was, just wanted to ask.
Is your question on what? So you asked what stimulus did we calculate the noise correlations themselves, and
right.
How does that? Because the ROI, the receptor fields themselves are, in your assessment, built up from noise correlations in the first place. I don't understand how the receptor fields are built up from noise correlations.
Can I reframe the question? I basically want to ask the same question, but on a different angle, which is
sure.
This receptive field overlap statistic, I think it would make sense to compute that cross-validated. We talked about cross-validating earlier. At least the receptive field overlap, if you cross-validate it, would be important to answer this. And then you can compute noise correlation by subtracting all receptive field properties from the cross-validated. Yeah.
So, but how does that relate to the noise correlations?
Because the noise in your measurements is contributing to your estimate of the receptive field. And so, by definition, two things with overlap had responses in a overlapping subset.
I see what you mean. So I need to control for the. I made some efforts to control for this, which I'll go into, but it's sort of becoming clear to me that maybe I needed to do a little bit more conceptual work to kind of produce a valid inference from this. Sure. I think someone else has a question as well.
More of a comment. I think that in a lot of these cases, you need to be careful and cross-validate or look for statistics which are not going to so much depend on tail of activity, because as Kaspar presented in here, I actually do think that all your conclusions could potentially be affected by this. So knowing the fact that you're going to have noise and you're going to have variability in there, you'd need to have a very clear quantitative answer for how much that is, what are the answers, because some of the conclusions in here can really be invalidated by that, by not doing the cross-validation.
Okay, thank you. I think that's something that I'll need to look into and potentially discuss further, is like how to perform this cross-validation in the first place. I'll try my best to present some controls that I put in place to sort of deal with this. But at least tentatively moving forward with this, I then looked at how RF and sort of relates to noise correlation in a sort of general sense. And what I found are these sort of like functional neighborhoods in RF space, where the higher the sort of receptive field similarity, the greater the noise correlation of those regions. And the sort of, this is just another chart to show that. And then sort of like splitting it up into these bins, we can kind of see this interesting thing that may go some way towards answering some of these questions, where if we look at these regions of RF similarity, they're like hugely distributed across the dendritic arbor. There's, at least sort of visually speaking, you can see that although these kind of segregate rather nicely into different bins of visual space, they're sort of distributed quite widely in ROI space, which implies that the sort of like it's the functional sort of input is the kind of structuring principle here, as opposed to topographic. I tried to do a sort of like within versus between bin sort of comparison, and we can see that our like this permutation analysis goes some way towards showing that this is a kind of significant finding. Of course, keeping in mind now that like maybe the inputs themselves need to be re-examined, and we need to sort of baseline subtract the noise correlation so as to, again, this is just
can I just clarify? So so is the hypothesis here is that noise correlations allow to sort of identify input domain that are coming from the same axons? Is that what you're thinking?
So what I was thinking, is it okay if I address that like in a couple of slides? Because I'm kind of trying to build towards my conclusion here.
Okay.
Which is, so we have this relationship between noise correlation and RF overlap. But we also know that a big contributor to this relationship is simple proximity on the dendrite, right? Like you would expect that to be the case. So the next thing I tried to do was control for physical distance on the synapse, on the dendrite, sorry. So I did kind of two measures here. The first was the partial R, where I tried to figure out kind of what proportion of the RF overlap, what proportion of the noise correlation is attributable to RF overlap, and what proportion is attributable to distance on the dendrite. And I try and remove the distance related part and ask whether there is the remaining RF overlap variance still tracks the remaining noise correlation variation. So the partial R's were all positive, or like mostly positive and significant. And then the far bin R is quite a crude kind of test where I only took ROIs that were on like very like distant ends of the synapse, or like furthest bins possible, and see if we still maintain this relationship between receptive field and noise correlation. And it seems to have sustained through this pretty well. I did a couple of other attempts to kind of validate this. One was a label shuffle, position label shuffle, and trowel shuffle, and split half as well. I can take any questions about this if anyone has any, but
it seems to be some form of cross validations, right?
Well, I maybe I didn't understand the terminology very well, but that's
right. That's right. I feel like cross validation is when you split the data in half.
Well, yeah. That's exactly what I did. Is I tried to destroy all temporal ordering, tried to destroy, or like anything that's contingent on, you know, the specific labels. And then over here, obviously, I try to maintain internal consistency of the data as well. I don't know if that actually addresses what was being asked, but and then I think it's also worth noting that the shuffled RF maps actually killed the effect, right? So there's at least some validity to some of this. So then the next thing I looked at was whether RF matched pairs co-fluctuate across distinct dendritic compartments. So now I picked overlapping sort of ROIs on DMD1 and DMD2 of each of these sort of different sessions. And I tried to see if we still maintained some noise correlation if they encode the same region of ROI space. And effects seemed to remain stable even when we only looked across planes. Yeah, I don't know if anyone has any questions on that point, but obviously the effects were somewhat weakened, but it still remained sort of reasonably significant here. You can see over here that, like, I think you lose 2 or 3 imaging planes depending on whether you look at mutual best enrichment, where I looked for the best matching RF partners, so 2 ROIs with the best matching RFs, and I called those mutual best. And the question I asked was whether the strongest cross-plane RF matches co-fluctuate more than ordinary cross-plane pairs. And it seems to be yes, modestly, they do. And at this point, I was sort of concerned that the ROIs themselves might be spurious. So I just tried to overlay it on the red channel. And we can kind of see reasonably well that a lot of these are plausible ROIs. The numbers obviously correspond, so this number 5 associates with this number 5 over here. Again, I think it would be really interesting to do this on the mean green channel if possible. That would be, you know, a good next thing to try. Thank you. Then I was concerned that potentially my Z threshold was too permissive or potentially too restrictive. So I reran the entire analysis across different Z thresholds, running from 2.5 to 4. And generally what we saw is that the effects strengthen the more sort of punishing the Z threshold is, which I kind of inferred from that the more sort of spatially selective an ROI is, the more noise correlated its input will be. But at least I was kind of glad to see that it doesn't seem to be like contingent on a particular Z threshold in the first place. And the sort of apologies for the kind of janky graphic here, but the sort of general thing that I, you know, kind of took away from this was there's probably an interestingly specific shared presynaptic population that's delivering rather specific synaptic drive to individual ROIs across compartments. And on some level, this may seem like a truism insofar as if 2 like localized regions of different dendrites are receiving input upon the same visuals, like the same RF, the same visual space as stimulated, you'd expect them to have relatively similar synaptic drive. But I just find it quite interesting, if we go all the way back to this slide over here, how distributed these are across the arbor. And that means that there must be at least some pretty interesting level of sophistication in the wiring here, that the same synaptic drive is delivered to synapses at seemingly arbitrary locations along the arbor. And then the question that I'm left with is, obviously we know that on the population level, we have pretty strong topography. We have orientation columns in carnivores, and even still some level of orientation topography for mice. So how are these sort of like analog inputs that seem to be like functionally localized over here? They seem to be sort of like functionally, like the functional input seems to be the organizing factor. How does that summate and integrate to form these topographic maps? Like, what is the dendritic computation which allows that to happen? And that's sort of the big question that I was left with at the end of this. There's a couple of questions, I think.
Can I just go?
Alex? Yes.
Yeah, so first of all, a beautiful work and very well presented, very nice work. Thank you. I have a very quick methodological question. So you say, so what data did you analyze for the noise correlations? Here it says static grading, but it actually shows the receptive field stimulus. Which
right, yeah, over here, this is a very janky graphic. Sorry, this is not methodological. This is very, this is just a suggestion of what could potentially be going on over here. This is a, yeah, this should be an image of a grating. It's not an image of grating. Apologies for that. But I just wanted to give some indication of what I'm sort of trying to indicate with this.
Well, it's important because some of the critique would be if you just analyze the receptive field data, then you would, that could explain a lot. But if you analyze the static grading data, that makes a big difference. And you're saying that's the one that you analyzed.
Wait, sorry, just to clarify, I only looked at the receptive field readouts. I didn't look at the grating, right?
Well, that's a confound, right? So then what you're actually looking at, so basically you're looking at variance in responses, but you're also looking at synapses that correspond.
Yes.
They're responding to the, so they respond in time. So that can already explain a lot of the noise correlations. Like what you actually want to do is you want to look at the data where the grading is presented and all of the synapses are responding at the same time. And then you want to show that those co-fluctuate or covariate and the rest doesn't. Does it make sense? So in other words, how can you rule out that the stimulus is driving a lot of this? Because you, in a way, you're just looking at trial differences rather than at the same time, these ones correlate and those don't.
No, I agree. I agree with you that to some level, the reason I didn't take it to be a confound is I agree that it's kind of trivial to say that the same stimulus will drive the same responses to all receiving ROIs. But that's almost kind of immaterial to the point that I'm trying to make, which is the fact that like these, even if I am saying that the same stimulus is driving the same sort of like the same synaptic drive, the destination of the synaptic drive is distributed across the arbor. So that's the first point, which I just find to be interesting in and of itself, which is it's not that for a given stimulus, the fact that the synaptic drive is distributed across the arbor, or whether I take all of the stimuli, then there's an additional question of the contextual modulation of whether or not changing the grating. I tried to look at that a little bit, but honestly, that part of my analysis is quite underdeveloped, which is why I didn't present it today. I'm trying to just present something very constrained, which is this relationship between sort of synaptic drive, which I may well just be tracking the stimulus input, but the fact that it's delivered in this sort of like distributed manner implies to me that it's the input that's structuring dendritic space, not dendritic space structuring the sort of receptive field, if that makes sense. It's the opposite of a classical topography is what I'm trying to kind of drive at with this idea over here. And then the second thing was, do you not think that at least some of this sequential problem is addressed by destroying the sort of like the temporal aspect by shuffling the trials and the positions?
Maybe I have to think about this a little more. I have a few concerns about it, but like, how about this one? Why not look into the baseline correlations and show that if there's no stimulus and you're just looking at spontaneous activity, you find the same pattern. So instead of noise correlations, you look at functional connectivity.
I think that would be a really good next step for me to do. Thank you for the suggestion.
I think using noise correlation as a way to pull ROIs together is an interesting idea. I don't know if it's used too much in literature, but it does have some base. Like you not only signal but correlation in fluctuations should be indicative of potentially share inputs. So I think that's a good idea. But
yeah, so
as Alex said,
maybe I can throw it in real quick. My concern is that you're conflating two things here. So as you're showing a particular stimulus, then yes, certain synapses will be activated and other ones are not. And so, probably that's because they get maybe axonal branches or similar kind of inputs. But it's also the whole system is in a different network state each time you present the stimulus, right? So it becomes difficult to dissociate these two. So I understand the logic that, well, what's even the difference there? But it would be cleaner if you do that as all dendrites are activated, because you show a full screen stimulus, a full screen grading, and then show that within these activated synapses, not the ones that are just activated at this moment, which could be for a variety of reasons, you do see that they co-fluctuate. So now it seems to me it's conflating which ones are active and which ones co-fluctuate. And in a way, it's like, well, the ones that co-fluctuate, that's the only ones that are active.
Yeah. I see your point. Thank you.
Manav, are you gonna post your presentations on the forum, or did you already, or what was your?
I'm happy to share it. That's I haven't yet.
If you were willing to share on the forum, I suspect that you get a lot of inputs on specific figures from this group, because the devil's is in the details, as everyone said, and we can continue on the forum, the conversation, and help you out.
Thank you.
It's 9:48. I don't know how long is Suraj a presentation. Do we have enough time for it?
I suspect longer than 10 minutes, but yeah.
Yeah. What's your preference, Sarah? Should we start there, or should we postpone it to next week?
I mean, maybe we could start at least some of the examples that she has in her analysis focus on orientation tuning to specific stimuli. And so it could be a bit of a segue regarding some of the questions we just had, basically. It's just that she does everything independently of the receptive field, and we kind of divided up the questions, basically.
But Soraya, are you open to continue next week?
So next week, 26. Yes,
I have exams, so that's the warning flag.
Yeah, that's the only thing.
Oh,
but I can
I'm sorry.
try to speed through it, or like present.
We can find you can start, and we can find another time that will work for you.
Okay.
Because I know you're prepared for it, and so appreciative of that.
Okay, I'll start. And then I'll carry on next time. Sorry. Can you see only the slides, right?
Oh, yeah,
or can you see the notes as well?
No notes. No.
That's
Okay.
right.
Okay, so
yes.
Sorry. Oh, my work was on experience-dependent plasticity of orientation tuning of synaptic inputs onto V1 primary visual cortex neurons. So I have started to explore experience-dependent plasticity on orientation selectivity of synaptic inputs by looking at the question, do oddball blocks cause lasting changes in orientation selectivity? So can I just minimize this? So to study the effects of oddball block 1, for example, I have compared it's quite slow. The effect of oddball block 1, for example, I have compared the orientation selectivity between the control tuning blocks 1 and 2, and for oddball block 2, tuning blocks 2 and 3. The aim was to build on the knowledge that orientation selectivity, that the orientation selected responses in V1 vary with stimulus context shown by stimulus-specific adaptation and deviance detection in the oddball paradigm from the Hammer-Tau 2021 study. And quick disclaimer that the imaging modalities, so for glutamate and calcium, and cell types such as pyramidal and BIP interneurons were pulled in a cross-subject analysis due to small subject numbers. And I have carried out the level of analysis I carried out started from examples of ROIs, so individual synaptic inputs. Then I moved on to ROIs in one dendrite, so ROI population within a session, then across neurons, so across different mice. So to begin with, to answer the question, does tuning strength and preferred orientation change across blocks? I plotted a polar plot for each ROI for tuning block comparisons separately for apical and proximal dendrites. So in blue, you can see the DFF activity strength in for the tuning block 1, and then in orange, you can see for tuning block 2. I used the vector sum method to extract the parameters tuning strength and preferred orientation from the Mazurak et al. 2014 study, like the paper. And then these were the formulas. And so the top row is for the apical dendrites. And then for the bottom row, it was proximal dendrites. The variable changes were also seen across tuning block 2 and tuning block 3 for both the components. This heterogeneity meant that warranted me to pull ROIs together and look for overall trends across ROIs. So then I wanted to, in addition to the tuning strength and preferred orientation, I wanted to find out does tuning sharpness measured in half width at half height change across blocks? And I've done this by applying a constrained single Gaussian fit method to extract this parameter, the tuning sharpness, and which basically involves applying a modeling curve onto each ROI's tuning curves, and then applying these formulas. Then the half width at half height was stated to be like reliable way to measure tuning sharpness, and from this paper. And the example shown here is just for the apical dendrite for tuning block 1 and tuning block 2. I just wanted to show examples of how the tuning sharpness varies. And this is seen for all dendrites and across all tuning blocks. And again, I wanted to analyze them together. I wanted to pull them together and do a population analysis to see the overall trends. So the next question was, how does tuning strength vary in apical dendrites at the ROI population level within a session? I plotted a histogram with all the ROI values. So for tuning block 1, this is for only one dendrite, the apical dendrite. So for tuning block 1 in blue, and orange represent the tuning block 2, like the distribution. And I've done this for again for tuning block 2 comparing with tuning block 3, and I found that mostly there was unchanged. The tuning strength wasn't changed, and but there was still a small decrease from tuning block 2 to tuning block 3, and I just thought that this might be due to cumulative stimulus-specific adaptation. And although the individual ROI changes were variable, they cancelled out at the mean. So when I applied this histogram to the proximal dendrite ROI population within a session, I found a very similar trend, a small decrease in tuning strength from tuning block 2 to tuning block 3, as you can see there. And again, the changes were variable and largely cancelled out at the mean. So for the tuning sharpness, I wanted to assess the tuning sharpness as well, and I plotted the histogram as well to visualize the data, and the distributions were mostly positively skewed and remained unchanged across tuning blocks. Same trend for the proximal dendrite. And I thought that the stability may reflect tuning sharpness being a fixed property of synaptic inputs, perhaps, and a positive skew likely arises from the constrained single Gaussian fit model, modeling perhaps two peaks, tuning curve as a single peak, and this might inflate with estimates. Then I looked at the preferred orientation and how this varied in apical dendrite at the ROI population level within the session. So still same level of analysis. And I found that with a scatter plot, that all the ROIs changed. So there was 100% change in ROIs, as this might be expected due to the vector sum method. And the circular distance were small, so 15 here. Sorry, circular distance was quite small for both of them, and I did the same for the proximal dendrite as well. But when I looked at the cross neurons, I found that in the apical dendrite, so every point here each point represents a mouse, a single mouse, a different mouse. And I've pulled, I haven't shown this, but I've done a comparison across sessions within the same mouse. Then I found the averages of those, and then these are all circular averages as well, and circular distances as well. So I've averaged them and then found the values for each mice and then compared them to each other and how they were all changing. And I did that for the different tuning blocks comparisons. And I found the largest change here. And there's supposed to be another point there behind this inset box, but the same pattern was seen. So basically, the preferred orientation changed a lot in the tuning block 2 compared with tuning block 3, compared to like tuning block 1 versus tuning block 2. And then I've done the same for the proximal dendrite to see if the same trend can be observed, and it was not showing the same trend, but there was no statistical significance in both dendrites due to small sample sizes, unfortunately. And I used one sample Wilcoxon on circular distances and circular correlation, and a Watson U-squared test, 2-sample Watson U-squared test to compare the circular averages. So the conclusion I was able to make, what I made, was that at the ROI population level, only preferred orientation changes were detectable, and tuning strength and sharpness changes were heterogeneous and cancelled out at the mean. This was consistent with G et al. 2010, who showed that afferent inputs with the same orientation preference are widely dispersed across the dendritic tree. So I expected heterogeneity in the preferred orientation, but I didn't really expect, it was not shown before that these synaptic inputs varied across blocks. At the neuronal level, the preferred orientation showed the largest change in pyramidal apical dendrites and superficial VIP somas. So I mentioned before that I grouped these two together, particularly between tuning block 2 and tuning block 3, and was absent in proximal dendrites and deep VIP-somas. So this may reflect compartment-specific input sources, with apical and superficial compartments receiving top-down feedback, and proximal and deep compartments receiving feed-forward input. The limitations of my study was that there was a small sample size, so this meant no Wilcoxon test could reach significance regardless of effect size. The modalities were also, and cell types were also pulled due to subject numbers. And additional finding, which I thought was quite interesting, was that the vector sum magnitude, which was a measure of the tuning strength, varied even within individual tuning blocks across all ROIs in both apical and proximal dendrites. So future analysis could test whether this within the tuning block activity predicts direction of change in ROI responses. And these are my acknowledgements. Thank you. Do you have any questions?
That was amazingly quick.
There are some really interesting nuggets in your presentation. I found the difference in orientation tuning change between the two compartments very interesting, and so given that we are getting more data, I think this is something to repeat on this rate. That will be interesting to see if it's confirmed. I think some of the groups also have found a similar thing. I recollect Nicolas presented along those lines in the past, so that seems to be supportive. But yeah, just wanted to highlight a few things. So yeah, thank you so much for this presentation, Stefan.
Mainly a comment. Fantastic presentation. I know you only had 10 minutes, but you were able to present really great results and also very well statistically backed up. So wanted to say fantastic work.
Thank you. I hope it was the plots were quite self-explainable as well, because I was just I was a bit stressed performing.
Yeah. One quick thing, when you try to do regressions, know that those measure errors only vertically. So it's not symmetric what you put on the X and Y axis. You can try to do a PCA, or you can try to measure a distance which is going to be orthogonal to the axis in order to not introduce a prefer. That was a tiny statistical comment because I can't keep my mouth shut, sorry. For everything. All the other tests going through and doing ordinal tests, I think it's fantastic, and it is always by far the most reliable. But of course, when you've got 4 or 5 samples, you can never actually even reach significance. So might try something which would be not ordinal or wait for more data.
Okay.
But awesome results.
Thank you. Yes,
Alex,
Alex, you have a question?
Yeah, actually, also just a comment real quick. I thought all your results were interesting, including what you showed that the sharpness and the width didn't change on the population level, but you did see it in individual ROIs. So I think that's worth actually keeping an eye on. I think that's interesting. It's kind of reminiscent of representational drift, but in a different way, right? So it's kind of, if you think about the brain, it maybe might just care that on this population level, the tuning width and strength is the same. But it accomplishes that by some kind of averaging, for example. And so we don't know. And so this data kind of seems to suggest that on a single level. So I at least am not aware of any work of that has been done like that. So I think even that finding is interesting. Obviously, even more interesting if you show it in combination with the changed tuning preference. But that side remark you did, I thought is by itself publishable and interesting.
Okay, thank you. If you want me to go back to any slides, then I can show you again if you want. But I'm sure I think we're running out of time, so I can also post them and the data as well.
Yeah, if you could post both of your presentations on the forum, that would sort of be helpful in the future. I'm sure we can return to it and also I think continue the discussion. I think Casper is suggesting to continue discussing the processing. He has some thought on to anchor with the raw data that we share. So I think we should continue on the forum. But thank you both for presenting. It was a wonderful, quick foray in the SLAP2 data, and there's clearly more to do, but thank you so much.
Thank you.
Bye.
Thank
Okay.
you.
Have a good day, everyone.
Thank you. Bye.
Have a good day. Bye.